Howdy, this is our gram acid fast capsule and endospore staining lab report for Group 2, Rita Cantu Mahurin and Diego Castillo. In our introduction, we really wanted to focus on the four key bacterial staining methods throughout this lab, which were the gram, acid fast, capsule, and endospore staining. Each of these are designed to differentiate microorganisms based on unique structural and chemical properties. And the purpose was to practice and compare the multiple staining techniques used to identify bacterial cell wall composition, morphology, and special features such as capsules and endospores. The materials that we used throughout this lab, including bacterial cultures, were the SRS, B. cereus, S. typhimurium, K. pneumoniae for the capsule staining, E. coli for the mixed culture control, Mycobacterium sp for acid-fast staining, and then the B. subtilis for endospore staining. For the media and slides, we used blood agar plates. We used prepared Mycobacterium slides. We also used TSB or tryptic soy broth, which is the mixed culture. And we also have the clean glass slides and wax pencils that we used. For the materials, we used staining reagents. So we used crystal violet, grams iodine, the decolorizer, which is alcohol or acetone, safranin. We also used the carbofusion acid alcohol decolorizer, methylene blue, malachite green, and copper sulfate 20%. Now in the procedure, you can see that depending on our purpose or the type of staining we were attempting to do, we didn't use all of the different staining dyes. So for example, the smear preparation, we used the broth culture. We transferred the loop full of bacteria onto a slide and we air dried it. We did use a hot plate to air dry it. From the agar plate, we added a drop of water. We mixed the bacteria, spread it thinly in the chalk circle, and air dried it. And then we heat fixed it only when specified, which was for the gram staining. In the gram staining, we applied the crystal violet for a minute and then we rinsed it with deionized water. We then added the grams iodine dye, which we rinsed after 2 minutes. And then we decolorized it briefly, immediately rinsed it with the DI water. And then we counterstained it with the safranin, which is for a minute, rinsed it and blot dried it. And then eventually you observe it under the microscope, hopefully under oil immersion. And then the gram positive was purple, the gram negative were pink. For the acid fast staining, we used the Mycobacterium prepared slide, and so we stained it with the carbofusion for 5 minutes and rinsed it. We decolorized with the acid alcohol and then rinsed, and then we counterstained it with the methylene blue this time for 4 minutes and rinsed and blotted. And then the capsule staining, we mixed the K. pneumoniae with the crystal violet. We dragged the mixture with another slide, air dried it only, and we didn't use heat fixation this time. And then we rinsed it gently with 20% copper sulfate blot, and then we viewed it under the microscope. And then for the endospore staining, we prepared the B. subtilis smear and then we used heat fixation. We stained it with the malachite green over steam for 7 minutes this time. And then we rinsed and counterstained with safranin for a minute. And then we were able to observe the endospores, the green, within the red vegetative cells.
These are our results for the gram staining. We have SRS, B. cereus, S. typhimurium, and our mixed culture. The first three appear to be as though they are burned or didn't heat fix enough, so you can't really tell the bacteria shape or color. For the SRS, it should have been a gram positive cockeye. For the B. cereus, it should have been gram positive as well, but a rod shape. For the S. typhimurium, it should have been gram negative and rod shaped. And for our mixed culture, well, it was a bunch of different bacteria that appear different, which you can kind of see in our staining, but it could have been better. In the gram staining, we used iodine, and iodine acts as a mordant, which will bind the crystal violet to form a larger complex that gets trapped inside of the thick peptidoglycan layer of gram-positive bacteria. So without it, we would have the crystal violet wash out during decolorization, which would make the gram positive cells appear as though they are gram negative, which would be inaccurate. These are unknowns in the gram staining, and you can see the unknown 112, see purple gram positive bacteria, and the unknown 292 is a gram negative rod-shaped bacteria as well. They are both rod-shaped, and the unknown 112 specifically shows us that it is in a chain-like fashion, while the 292 appears to be more of scattered, singular rod shapes. Um, Gram-positive tells us that it has a thick peptidoglycan layer that'll retain the crystal violet iodine complex, making it a purple color. And the gram-negative bacteria will have a thinner peptidoglycan layer and an outer membrane, so the complex is washed away during the decolorization, leaving that pink-red counterstain with the safranin. For the acid-fast staining, we had our negative, which was Corynebacterium spp, and the positive M. gordone. And for the acid-fast staining, we should have seen the positive appear as though it was a bright red or fuchsia rods, which we can see the bright red color and the shape somewhat. And for the non-acid fast bacteria, it would appear blue, and it would be because they were decolorized by the acid alcohol and will take up the methylene blue counterstain. And the positive would retain that carbonyl fuchsin stain due to that waxy lipid-rich cell wall. And this one used the decolorizer as well. And the decolorizer is important for removing the crystal violet from gram-negative cells when we're talking about the gram staining. But for acid-fast staining, it's important in removing the carbonylfusion from non-acid-fast cells, and then that allows us to counterstain them with the methylene blue. For our capsule staining, we have arcane ammonia with the crystal violet drop and arcane ammonia with the deionized water drop. These came out really good. In the crystal violet drop, you can see the white specks, which are where the bacteria is that didn't get stained. And then the right, we see our actual capsules, which are the cockeye-shaped bacteria that we see. And in capsule staining, we use copper sulfate, which is both a decolorizer as well as a counterstain. It'll remove excess crystal violet without rinsing away the actual capsule. And it also lightly stains the background, making the capsule appear as a clear halo around the stained bacterial cell. And it's important to air dry it and not heat fix it in capsule staining because it can destroy or distort the capsule because it's so delicate and non-ionic. Air drying preserves the capsule structure, allowing it to be visualized as a clear halo around the cell. For our endospore training for the B subtilis, it came out really well. You can see the red bacteria with the blue endospores inside of them, and it came out really great. We used two methods. For ours, we used one where we steam fixed and one where we heat fixed it normally. And this is the heat fixed one without the steam fixing, and it came out really well. Um, and the purpose of that steam fixing, though, is to drive the malachite green into the tough spore coat of these endospores. Without the steam, the dye wouldn't penetrate, and endospores would remain unstained or faint, even though for us, we can really still see it. That's would happen normally if we didn't steam it with the steam. But when we do steam it, the slide is counterstained with the safranin that highlights those vegetative cells in red, while the endospores appear that green-blue color that we can see here. Okay. In conclusion, the lab demonstrated 4 bacterial staining techniques: gram staining, acid-fast staining, capsule staining, and endospore staining. These were used to differentiate microorganisms based on structure and composition. Gram staining separated gram-positive and gram-negative bacteria from each other. Gram-positive would appear as purple, and gram-negative would appear as pink. Acid-fast staining, highlighted by Mycobacterium species, would show a positive as a pink color because of the waxy acid-fast cell wall, and the negative as a purple color, or blue. Capsule stain showed the cane ammonia as clear halos for the capsules when using the crystal violet, and for using the deionized water, you could see a just purple dot for where the bacteria is. In the endospore staining, we identified green endospores within the B subtilis, and you could see them throughout the different bacteria, showing the endospores. These techniques were essential for bacteria identification, classification, and understanding the pathogenic features of the bacteria. They also helped us identify what kind of gram uh, stain the unknown bacteria we had were, whether they were gram positive or negative.